Hello everyone, welcome to chemistryATLAS. Uh, Today in this video, we'll be talking about the other forms of hemoglobin, that is hematin, methemoglobin, and we'll also be talking about the cyanide poisoning. But before proceeding, in the previous video, we have missed out some points on hemoglobin. So let us look at it. When hemoglobin binds with oxygen, it shows a Raman stretching frequency at 1100 centimeter inverse. Okay, this is important and you must remember this one. Then next we have the Bohr effect. Bohr effect. Bohr's effect tells us about the binding of oxygen with hemoglobin, which is dependent on the pH of the blood, If pH decreases, binding affinity of oxygen with hemoglobin decreases, and if pH increases, the binding affinity increases. This Bohr's effect is also dependent on the concentration of carbon dioxide. Carbon dioxide reacts with water to form carbonic acid, which results in an increase of carbon dioxide, resulting in the decrease of blood pH. Hence, hemoglobin proteins release the load of oxygen and conversely, a decrease in the carbon dioxide contents provokes an increase in the pH level, which results in hemoglobin picking up of more and more oxygen. So let's start with our today's topic, hematin. Hematin is also known as naked heme, oxyhemochromosome, or oxyheme, etc. So this is generally a heme protein without any protein chain attached to it. So it becomes easily susceptible for oxidation by the surrounding water molecules by the surrounding water molecules present in the blood. So let us look at the mechanism of how a hematin results. So this is the porphyrin ring comprising of Fe2 plus. So when oxygen binds to the ferrous ion, it converts to, it is linked in a band fashion, or we can write this one as also like this. So this one, when it combines with again one more ferrous ion, it converts to a mu peroxo species. So mu peroxo species of ferric ion. Then homolytic cleavage takes place, resulting in two ferric radicals, which gets converted to a ferryle complex. This is a ferryle complex, and finally when it interacts with one more ferrous ion, it gets converted, which is a mu-oxo-dimer of ferric ions. This hematin is a dark bluish or brownish pigment containing iron in the ferric state obtained by the oxidation of heme. Next, we will be talking about methemoglobin. So, it is an oxidized form of hemoglobin, that is, the iron is present in +3 oxidation state, so it does not take part in oxygen transport in our body. So next, how a methemoglobin is formed. The hemoglobin, which is bound to the protein chain, does not allow oxidation of the iron center by the surrounding water molecules present in the blood, but then also 3% of the hemoglobin gets oxidized to methemoglobin. In order to overcome this problem, there is an enzyme known as methemoglobin reductase, which converts back the ferric ion back to the ferrous ion of the normal form of hemoglobin. That is, hemoglobin, almost 3% of the hemoglobin gets oxidized to methemoglobin, which is again reduced to hemoglobin in presence of the enzyme methemoglobin reductase. However, in absence of this enzyme, if the concentration of methemoglobin increases, This leads to a disease. This leads to a disease, methemoglobinemia. This methemoglobin also plays an important role in preventing the cyanide poisoning. Now, we'll talk about cyanide poisoning. So, if we talk about the normal electron transport chain which takes place from cytochrome B to cytochrome C, followed by electron transfer to cytochrome A, and finally electron transfer takes place to oxygen, giving a peroxide species which then reacts with two protons, resulting in water and energy. So in presence of cyanide, what happens is that the cyanide binds with cytochrome A and electron flow to oxygen is stopped, due to which water and energy is not generated, and cell death cell death takes place. That is, cyanide binds with cytochrome A, okay, hindering the electron flow to oxygen, as a result of which this water and energy cannot be produced further. So in order to overcome this problem, a solution of sodium nitrite is inhaled, which oxidizes ferrous center of heme to Fe3 positive, that is a methemoglobin. The cyanide which was binded to cytochrome A now binds with Fe3 plus of methemoglobin and gets excreted through urine. Then the electron flow again starts without any hindrance, and this is how methemoglobin can be used in the treatment of cyanide poisoning. So this is all for the today's video. I hope everyone of you like this video. Please hit a like, share and subscribe.